Where is the camera?
Or
I don't know what we're doing here, but, oh, that's the camera. Welcome everybody in person and those of you who are doing this online. Today, we have our seminar by Francisco Menendez. It's my pleasure to introduce him. I'm his primary advisor. For those of you who don't know me, my name is Ciela Maximova. I am still a faculty in the Department of Plant Sciences. And it is my pleasure to introduce Francisco. He came to us about 4 years ago. He is currently a graduate student in the plant biology program. He's originally from Peru, and he completed his undergraduate degree in biology from University Heredia in Lima, Peru. He had an interesting path before he came to us. He worked for a consulting agency in the Smithsonian Institution. And so now he's going to talk to us about a issue that is really important for cacao. Francisco is part of the Penn State biology and the molecular, Penn State endowed program of the molecular biology of cacao. And so, go ahead, take it away, Francisco.
thank you very much, Ciela, and good afternoon to everyone here joining us in person. This is my first plant science seminar. I'm very excited, and welcome to everyone else online as well. So, as Ciela mentioned, my name is Francisco Menendez Burns, and today in this seminar we'll be talking about the characterization of cadmium transporting activity of a candidate HMA gene of Theobroma cacao. Okay, so first I will start with an outline of my presentation, and I will begin by beginning a background on the crop, Theobroma cacao. Then I will give a short background on the process of metal uptake in plants. I will present the scientific questions that I want to ask, and also the approaches I will take to answer them. I will talk about the impact of cadmium on the cacao supply chain. Then we will go on to some results, the experiments that are still in progress, some conclusions and summary, and finally some conclusions and summary. So, Theobroma cacao. I'm sure everyone here have had chocolate before. Theobroma cacao is a source of the main ingredient in chocolate, cocoa beans. And it's a tropical tree that evolved in the Amazon River Basin and now is grown almost in all humid tropical areas. And it provides livelihood, or at least partial livelihood, to many small farmers. And here I'm showing you guys a picture of this agroforestry system that kind of is where cacao is grown. This particular picture is in Cusco in Peru, a little experimental plot. So unfortunately, the main ingredient in this delightful food, chocolate, is in a vulnerable position. The crop not only suffers many challenges from disease to climate change. Now, more recently, it's subject to regulations that limit the amount of cadmium allowed in the products that we consume. And here, if anyone has not seen a cocoa pod before, the one on the bottom, you can see a cocoa pod here. And it's a hard shell with a sweet, like, kind of mucilage surrounding these beans that then get fermented and produce cocoa. And here on the right, we have a chart of the proportion of production worldwide by country. And I just want to make a note here that up until 2016, over 75% of the world cocoa production happened in Africa. And then followed by that, you have Brazil, Ecuador, Peru, Dominican Republic, and the rest of the world. But you can see the lion's share goes to Africa. That's where most of the chocolate that we consume comes from. Okay, so cadmium and cacao. Why is it an issue? So many of you may not know, but exposure to cadmium, either chronic or acutely to a high concentration, is toxic, and in many cases, and extreme cases, can lead to kidney damage, bone diseases, osteomalacia, types of osteoporosis, demineralization of bones, and even cancer. Now, cadmium is present in soil, and usually it's more frequently found in volcanic origin, soils with volcanic origin. And plants in general uptake cadmium from the soil and then store it in different tissues. And they have like different mechanisms, and there's variability. Some are hyperaccumulators, some are not. I'm going to talk a little bit about that later. But cacao also does that. And after a wide survey of a lot of different types of food, the European Union decided that they would impose a regulation that limits the amount of cadmium allowed in chocolate and chocolate-derived confectionery products. And you can see here, I have a little screenshot of the title of this document. If you are interested, it was actually published in 2006, but the regulation came into place in 2019. And farmers from Latin America, which is the center of origin of the species, are the most affected as the continent has a recent volcanic origin. So cadmium and plants, what is it doing there? Why is it get there, right? So cadmium is also toxic to plants, and it's a non-essential nutrient or element. And I say non-essential because to this day, we haven't found any function that it would fulfill. So why do the plants uptake and store this element? Cadmium mimics or is physicochemically similar to zinc and iron, which are essential nutrients. And this plant, you would say, mimics zinc and iron and utilizes, because it has divalent positive cations, it's a divalent positive cation. So it uses the same pathway that zinc and iron would use and to get into the plant. We're putting this like consciousness on the cadmium, but it just happens to be the case that it uses these other pathways. Now, abiotic factors that may influence cadmium uptake. Number one is the concentration of cadmium in the soil. That is the biggest factor for by a long shot. And in terms of mitigation, if any one of you here or online is trying to start a cacao farm, make sure that it does not have cadmium in the soil. That's the biggest prevention that you can do, right? Make sure that before you start, it's a perennial crop. So now, if the case is that you already started your crop and you find that it has cadmium, soil pH can also have a strong influence on the uptake of cadmium. Mostly because the pH, at lower pH, cadmium becomes it turns into an ionic form in a cation where it is actually soluble in water. So that is when it's actually becomes available for the plant to uptake. At higher pHs, this can change. There is a mitigation research done with soil amendments that change the pH. But the success is really limited to the initial topsoil, so not as efficient. And then another factor that influences cadmium in abiotic factor is soil organic matter content. So how much organic matter is in the soil? Organic matter usually can be negatively charged. That would trap positively charged ions. And then finally, if you have a zinc or calcium deficiency in the soil, that can also affect, because as we mentioned, they use the same pathways to get introduced into the plant. Now, usually the concentration of cadmium is way lower, so it has to compete for those entryways. If there is a deficiency in zinc or calcium, usually there is not as much competition, and it's easier for cadmium to be stored and uptake by the plant. Now, biotic factors that influence cadmium uptake. I think the microbiome is something that is being researched right now and could play a huge role in cadmium in the rhizosphere. Also, the cacao production systems that people use. So, like more in an agronomic level, if they remove the litter, if they have other companion plants in their agroforestry system, that can also change the amount of cadmium available. And then plant genetics, which is why what I'm studying was my interest. And fortunately for me, there is limited knowledge for on cadmium genetics and associated gene functions in cacao, unfortunately for the value chain. Now, just to put it in a perspective, the genetic variation of cadmium uptake in cacao, you know, it's been studied before, kind of recently. The most important takeaway that I want to give from this slide is that cacao on its own is not a hybrid accumulator. So we're not looking at a plant that's going to be like taking up the stored cadmium in a crazy amount. It's just it happens to be, you know, grown in sometimes cadmium concentrated soils. So I mean, usually, just as a reference, it wouldn't exceed sorry, I haven't even moved this. Sorry, it wouldn't exceed 10 ppm parts per million cadmium, which is milligrams per kilogram. And that's like at the high end in Latin American farms, really, really high end. Usually you find it like either undetectable or, you know, 1 part per million. The cadmium uptake, like I mentioned, has been studied before and does vary within the species. And interestingly enough, the correlation between cadmium stored in the leaves versus cadmium stored in the beans is actually not high, which tells you that the mechanisms are potentially very different for loading into the leaves versus the bean. And that's an important note. Whereas obviously cadmium in the soil is highly correlated to cadmium in the leaves. Okay, so going on to our overview of metal uptake in plants. Here I'm showing you a transverse section of a root in a schematic like cartoon representation. So just to get a notion, all nutrient uptake starts with water moving towards the center of the root by action of mass flow, and that carries solutes. Now, when we're looking here at this schematic here, let's say this is a little cation here, this gray thing. And once it comes into contact from the rhizosphere into the root system, it could first enter the apoplast. And that's what we're looking at in this blue line here. And if it travels through the apoplast, it eventually is going to land or be stopped by a gas barrier strip. It will not get into the steel. The steel, or the vascular tissue, is very highly regulated in what it emits into it. Now, if it doesn't get trapped by a negatively charged particle in the cell wall, it could also be introduced into the cell, and therefore the symplast, by a transporter such as NRAM, a zip type of protein, or a cation exchange protein. When it travels down through the symplast towards the steel, it can also arrive to a vacuole where it will be introduced for storage, for later use, or just kept there. And if that doesn't happen, it can actually go and arrive across the casparin strip next to the xylem parenchyma, where by the activity of another extrusion protein, such as HMA2 or even IRT1, can pump these ions into the steel, and once it's in there, it travels through the plant in the vascular tissue. Now, I did mention the HMA family of proteins, so we're going to talk about that in this presentation. So here, I'm going to show you 3 points where HMAs might be involved. And this is data that initially was produced in Arabidopsis. So the first protein that I want to mention is the vacuole sequestration one, and that's an HMA3 type protein. This HMA3 in Arabidopsis will reduce the amount of cadmium that gets moved up to the aerial parts of the plant. And then we have HMA2 or 4, which are xylem loaders, and they will basically be responsible for delivering any nutrients into the steel, and mainly zinc. So these mutants of these HMA2 and 4, which both are redundant between them, are very zinc deficient. And then finally, seed development, it Also, HMA2 and 4 participates in moving nutrients from the outer layers of the seed coat, into the cotyledon from the mother tissue into the filial tissue. Is required for seed development. Zinc is very important. So I'm just showing you here, this is how an HMA protein kind of would look if we would like to do a cartoon schematic. It has, I want to mention just that it has these conserved domains, the A, P, and N, and they serve very specific functions that we're going to discuss a little bit. And they have these transmembrane sections. And cadmium, they form a little tube and cadmium comes in. And how it works, this is a very high efficiency transporter. So it would it has two states, open and closed. In the open state, it will accept a cadmium molecule into the coordination site and also an ATP molecule. Once it's in that, both are connected, it hydrolyzes it phosphorylates one of the sections and then changes the conformation, releases the cation to another side of the membrane, and then returns to its open state, ready to do another cycle of pumping. So just to get an idea how well conserved these proteins are, I'm showing you here an alignment of mainly plant species. We have two from cacao that we're gonna be studying, we're gonna be looking at later, but also Arabidopsis halleri, which is the hyperaccumulator version of Arabidopsis, so to speak. We have some rice here, proteins. And then finally we have, I want to highlight these two, Staphylococcus aureus and Bacillus subtilis, which are two prokaryotes that still, if you see here, these bar charts, very high similarity. There's some variation, but here they have 100, the green means 100% similarity across. And, in the N domain and the P domain, this is a very important domain for receiving the phosphate group. That here, even in Bacillus subtilis and Staphylococcus aureus, is conserved. So these are canonical domains in this type of proteins. Now, here, now obviously from there I went to recollect the HMA proteins in Arabidopsis, and there are 8 HMA proteins in Arabidopsis. And I'm showing you here this kind of loaded slide of this table. But what I want you to concentrate here is, I already mentioned here's a gene name, and then here is a column that shows a substrate affinity. These can be classified by their substrate affinity. I'm interested in these two rows here that have cadmium in them and zinc and cadmium. So these here are the ones that are potentially involved in cadmium transport. Now HMA1 localizes, here we have the localization column, localizes to the chloroplast envelope, and it's thought to have a function in transporting copper into the chloroplast. Copper is an essential element for photosynthesis. And below that, we have these other cadmium transporters that are localized to the plasma membrane. So that's very important because it will potentially be responsible for moving ions out into the apoplasm we see necessary for nutrition, zinc nutrition. And then another one that's HMA3 that localizes to the vacuolar membrane, and that's in order to reduce the toxic effect of elements like cations in the cell. Okay, so we're going to focus on HMA2, 4, and 3 in Arabidopsis. I'm just going to, I have these references here, but I just want to say that HMA2 and 4 are zinc loaders with cadmium transport capacity. So you would imagine that these play a very important role in zinc nutrition for the plants. And they not only localize in the roots, but also expressed in the seeds. And then Arabidopsis HMA3, which is that vacuolar sequestration protein. And previously, they found that in Arabidopsis, soybean, and maize, mutants of HMA3 have a higher accumulation of cadmium or transfer of cadmium into the air or parts of the seed of the plant. Sorry. So that's an important, potential target for improvement of this trait. So my research questions for this project were, first, what are the orthologs of the HMA proteins in cacao? And then what is their evolutionary history? what biochemical functions do they have, and finally, what physiological functions do they have. So my general research approach will be obtaining the coding sequences from Arabidopsis thaliana, identifying the orthologs in cacao, studying their evolution, and we're going to do that through phylogenetics, study their gene expression, and then study the function in heterologous systems, namely yeast and Arabidopsis. So showing you some of the results that I have today. After proceeding, identifying and finding the orthologs, I identified 8 HMA ortholog orthologous proteins in cacao. Here we have the list of them. They all belong to this. But when I say orthologous, I mean that they came from the same. They were inherited by descent, and they didn't appear by a duplication after speciation. So they all come from the same ancestor, right? So these proteins all come from technically the same ancestor, the ortho group, And I proceeded to perform some predictions on these. One of them that I think is really interesting is the localization prediction, and I used this heuristic software, online software called DeepLock, and I recommend it. Really, it seems to be really accurate. And then you have transmembrane domain prediction using THMM2, which is predicts transmembranes. Now, if we look at the localization here, you got some that are going to the plastid, lysosome, another one to the plastid, mitochondrion. And then we have these that go to the cell membrane. Those are particularly interesting to me. Why? Because they have the potential to be cadmium or zinc extruders into the apoplast or zinc loaders into the atom, right? It has to be, otherwise they would localize somewhere differently. And so yeah, here is just the output of the DHM. Here you can see the predicted transmembrane regions in one of the proteins. Like kind of tells you what's inside and outside. Just an interesting to show you guys how that works. So focusing on these 2 cell membrane localized proteins. In 2001, Michael Palmgreen, one of the main researchers that worked on Arabidopsis HMAs, he made like this type of unrooted phylogeny and was able to classify the Arabidopsis proteins by their substrate specificity. In this tree here, you have 2 colors, one corresponding to substrates zinc, cadmium, lead, and cobalt, as he originally defined. And then you have on the other side, copper and silver. Now, we were talking about HMA3, 2, and 4 from cacao that are responsible for cadmium. And here, you can see them here. These are the 3 from Arabidopsis. And kind of as predicted, as we saw, the 2 of the ones that localize, of the 3 that localize to the cell membrane of cacao are here in the zinc, cadmium, and lead, cobalt area. Okay, so after that, I asked the question, okay, let's look into the evolution. Namely, we're finding 3 in Arabidopsis and 2 in cacao. This is a huge tree, and I don't want to go into the detail here. That's why I'm going to zoom into this area, which only shows the core eudicopsid cell group. I want to highlight here the Arabidopsis proteins. They all the proteins from one species grouped together, and that happens in all of the species. So, and even in the ones from cacao that are marked here with these black dots. Why do Arabidopsis have 3 and cacao has 2? It seems here, since all these seem to have at least, from 1 to 4 copies of this version of this protein, and they're all more related between each other than the ones that are in different species, that they inherited these from a common ancestor, these either 4 proteins, and then potentially lost them. And that might have been the case with cacao, where it inherited potentially more HMA proteins from this ortho group, and then lost them and kept these 2. But this is like still, kind of in the works, but that's one potential explanation. Now, when I decided to go ahead and do the functional experiments, I had to make a choice on which one to clone, right? And I just want to make that clarify that because these two proteins that we were talking about, they lie very closely in tandem in the same chromosome. Now and not only that, they also share 98% of sequence similarity at the coding sequence level. So we're looking at two very similar, almost the same protein, and cloning those was kind of differentiating between one and the other. It's kind of hard. So once I obtained one, and here you have this is the Criollo genome, which is one of the initial genomes that was published. This is the version 2. In the chromosome, and you have one here and another one here, right on top of them. And this is another genotype of cacao called Carina 6. And again, you have the chromosome here, and you got them in tandem. Now, the distance between them is way different, but these are genomes at this. So once I obtained one of these clones that was completely similar or identical to the coding sequences that we find in the Criollo version, I proceeded to, and then before that, so this is the one I decided to go ahead and study because I already had a clone of that one. So now I'm going to present a little bit about the expression of this HMA gene during development. I'm mining this data set that is the Gene Atlas. This has been produced by the Gene Atlas Consortium in our lab, and it's basically development of the expression of all the cacao genes during development in different tissues. So we know the gene that I was looking at, and we know that it potentially has a function in the root and potentially has a function in the seed. And here I'm showing you the expression, the absolute level of expression in fruit and the floral developmental stages, which is very important for pollen production. Zinc is very important for pollen production, and in the fruit developmental stages. So these, you can see that it's relatively high expressed. So it's a protein that's present during fruit and seed development. Moving along, if you look at the seedling stage, when it's kind of just starting, there's not a lot of expression of this. It's still, relatively present. But once it starts to have roots, you can really see that it's really highly expressed in the root system. And that trend continues further on, 3 months down the road. You can still see it has very strong expression. It's potentially, if we kind of imagine it serving the same function that HMA2 and 3 and 4, it might be, loading and delivering nutrients to the xylem. And 6 months down the road, it's still going strong. So it's very strongly expressed on the roots. And together with Lena here, we were able to clone it from the roots and the leaves. Okay, so moving on, after we saw that and we obtained that clone from the roots and the leaves, we proceed to study the biochemical functions. And to do that, my experimental approach is to use a yeast expression system. Now, yeast is a model system for functional genomics. It has a lot of known mutants for middle uptake, which is really great for what I'm looking at. And also, obviously, it's a eukaryotic system, which is kind of important for a plant protein, which is also a eukaryotic system. And it's easily transformable. So we proceeded to transgenically complement the mutants of the yeast mutants with this cacao gene. Okay, so just for the molecular biology people here, this is the plasmid that I chose to use, and it's called PYES2, Yeast Expression System 2, and it's under control of the galactose promoter, so I can turn it on and off, just neat. And and then here's the coding sequence of the gene I cloned from cacao. Here it is. Okay, so the yeast assay in itself, we have two main predicted scenarios in this one. One of them is that this protein causes a net ingress of cadmium mediated by the HMA protein, right? So here we have a very, very reduced version of a yeast cell. and you have this HMA protein on the membrane, and it's like obviously I say net ingress because there's movement outwards and inwards. The yeast cell has its defense coping mechanisms, but in general, if there's this pump moving in efficiently, it's going to produce an ingress in net, and that would turn into a reduced yeast tolerance to cadmium and a reduced growth. So we'd see less growth in the yeast. It would affect it negatively. And then scenario B, you have a net egress of cadmium that is mediated by the HMA protein. And basically, the idea is that it's, by any means, the cadmium gets into the cell, be it by its natural means. And this HMA protein is going to be it's showing its activity, moving out the cadmium, and therefore increasing the tolerance of the yeast to cadmium when I grow them in cadmium. So that being said, okay, I want to show you the results here. So here are the results of the yeast assay, and we have I'm going to go explain this step by step, but basically we have this is a yeast liquid culture that was placed on top of a media plate that contains either 0, 5 micromolar, 10 micromolar, or 20 micromolar of cadmium. And it was done with a yeast that contained what we're mentioning here as the empty vector, and you can read it here, but basically a yeast with a PS without an gene insert. And then on the lower row, we have the same yeast with my PS and the HMA gene insert in there, and they're all being expressed. This is growing galactose. So you have with the gene, with the HMA, and without. And at 0 micromolar cadmium, you can see that the growth, sorry, but this, further mentioned, these are serially diluted. This is the same culture, but I just diluted it in order to see the growth a little bit better. So at 0, you can see that with or without, it's pretty comparable. It's, doesn't seem to have a real strong effect on the growth of the yeast. Now, immediately after you grow it, if you add a little bit of cadmium to the media, you can see the effect of this HMA expression is strong, and it ends up reducing the amount of growth in the yeast cell. It's super strong. At 10 micromolar, you can barely see it, and on 20, it's almost everything but gone. So it's basically eliminating any type of growth from the yeast. So the main conclusion that we can get from this is that the HMA protein had a reduction in yeast growth rate in a cadmium dependent manner. And that to me signals one thing, that this protein in fact does have the capacity to move cadmium across membranes. So that's done for this one gene, right? So now I want to talk a little bit about the in-progress experiments, which I haven't, next steps for this research project is testing that same protein in a Arabidopsis HMA2 and 4 knockout and an HMA3 knockout. And we're going to see if we can rescue that phenotype with this cacao protein. And then we're going to complete the search for variants in the diversity panel, and we're going to look into that a little bit. But we have a panel of 38 sequence genotypes from cacao, and we're gonna see if there's variation between the genotypes, which I think there will be. Now, and then we're gonna observe the effect of cadmium on transcription, and for this we have a collaboration with the Colombian Corporation of Agronomic Research, Corpoiga, and that's also ongoing research that we're gonna be able to see what is the effect of cadmium on the cacao plant and gene expression. So showing you a little bit of the variation of the HMA protein in cacao in this in a reduced panel of 34 genotypes. So this is a protein alignment, and here we're showing I know it's a little bit hard to see, but this is an alignment And at the top, you have the same bar chart that I mentioned. Green means 100% similarity, whereas yellow means 70 to less than 100% similarity across all the nucleotides. So yeah, so I think the main takeaways that I can give from this piece of data is that the gene number across cacao varied from 1 to 3. So there seems to be copy number variation in the species between these genotypes. And there's a lot of mutations and deletions, but they're not frame shifting. So they still produce complete proteins. And then there's many SNPs which may or may not affect, the transfer capacity. That is still to be seen, but they're there and available. So yeah. So with that, I would like to acknowledge some of the people that helped me through this research. First, the Giltman Maximova Lab, Ciela, my main advisor, Mark, my co-advisor. I want to thank Lena and the past member Trinity Ham for helping in the cloning steps and getting my spurs, so to speak, in the lab. She still is, helping me out. I want to mention Akiva, Sarah. They were the main producers of the Gene Atlas. And then my advisor, my committee member, Claude de Pamplis, Eric Connolly, and Gabrielle Munchausen, who all helped me form this research project kind of from the beginning. Then obviously Isaac, who works in a similar project, and he's been super helpful in methods, and he's done a lot more in yeast as well. And finally, funding. I received funding from the plant biology program, from the plant science department, so I'm very grateful for them as well. So with that, thank you for your attention, and now I will take questions. Yes, Liana, please.
Okay. Yeah, that was great. Thanks. So I want to ask you about the gene expression pattern that you found.
Yeah.
And I guess I'm curious because when you set it up with the suggestion that there was a lack of correlation between accumulation in the fruit and in the leaves, if I remember correctly. But then when you were showing your expression data of these genes in cacao, it seemed like it was expressed both in leaves and in fruits equally. And I guess, do you have any thoughts about what's going on there, or what?
Yeah, no, that's a good point. I think that the main factor that might be contributing to that is storage, because expression of that gene, potentially could serve as a xylem loader, can be pumping stuff out, and mobilizing stuff out, or it could be storing it in the vacuole. So, I mean, I'm not sure if this one gene is responsible for all the variation between leaf and bean accumulation, for sure. That's, I mean, the data from the field is pretty clear, that the correlation is low. So it's like under 40%, 30%, so it's like less than flipping a coin if you're gonna get any good predictions. So I'm not sure why is that if necessarily this gene is solely responsible. I feel like that, as best I can say right now. I'm not sure.
So you're thinking there might be another gene that's actually underlying where it gets stored and how it gets stored that might be contributing to that.
I think expression of this protein doesn't necessarily equate to storage. That's another component, right? So it's mobilizing, but storage on its own can be mediated by a different protein.
So you have a question online from Claude.
Yes, Claude.
Yeah, when the yeast knockout is recovered by the cacao gene, what will determine if it plays a role in ingress or egress? And the second part of the question is, do independent recoveries behave the same way?
Yeah, that's a good question. To be completely honest, I don't have data on whether the yeast cells are accumulating, cadmium in them, right? what I can say is that, for one, there is a cadmium dependent reduction, right? Because when you see, and I can go back to that slide, when you see that when it's grown without cadmium, it grows in a comparable manner. And I've done this experiment a few times, so it happens quite a bit. Like, it's not something that I saw once. It's replicated. Now, whether it's a great question, because you come to think about it. Okay, if it is actually moving cadmium into the cell, you should be able to quantify that, in some liquid media, but it's also reducing the growth rate. So that's kind of a challenge for this. So can you repeat the question? I think I'm missing one part.
So what did you know about the yeast knockout recovered by the cacao gene, and what determines if it plays a role in ingress or egress?
Yeah, I'm not so. With this all kind of based on like our pre-made assumptions, right? So I'm based on this idea that it's localized to the membrane, right? And if it's pumping towards the cytosol, then it would have to have, a net ingress. To answer your question more accurately, if the cells are dying, as we see here, it means that they have more cadmium in them, right? That's the main takeaway. So in order for that to happen, it probably has to be facing inwards and pumping cadmium in. Now, I thought about this a little bit further, and okay, these proteins are ATP dependent, right? So how can it work without ATP if it's outside in the media? Yeast cells do export ATP out into their growth media, right? So that's one thing that I found. So it's something that yeast cells would provide to the protein, ATP, and the fact that it is dying kind of signals or points towards the fact that it is moving cadmium inside.
So Aaron suggested that you could do an ICP MS on those cells.
That's a great point. Yes, I know Zach here is working on something like that. So that's something to look into. I would have to titer the amount of cadmium that they're tolerant to, but yes.
And Surinder wants to know if that HMA also transports zinc.
I haven't been able to test that. That's a great question. If this HMA protein transports zinc, I'm still refining the conditions in which this zinc deficient yeast would be able to grow and express this HMA. Previous tests, I wasn't able to have any growth from this zinc deficient yeast, and I think it's more not to do with the protein itself, but the fact that I was growing it in its galactose media, and it might have some other deficiencies that I have to really nail in to achieve. But yeah, that's next, also in the works. Great question.
So Claude states if you get some with higher internal cadmium dying, while others accumulate less and do better, then maybe you could have a way to create a resistant cell. And Aaron mentioned that you could compete the cadmium sensitivity with increasing zinc.
That's a great suggestion. Take note of that. So
yeah,
Francisco, I have a question.
you could just say, with your 5 micromolar cadmium, you could just add increasing amounts of zinc and compete away the cadmium sensitivity. It's really easy to do that.
yeah, no, yeah, that's definitely. Yeah, I still have to work on that, and I'll perform that titer, see if we can get my ZRT1 C deficient cells to grow properly in the media before I try to express this HMA protein in there. But yeah, no, that's a great suggestion. Thank you.
So Surinder has another question. I'm going to unmute again so maybe you can hear him. Yeah, too much feedback.
I'm not doing that.
Type it into the chat box if we're getting too much feedback.
Should I ask my question, Francisco? So my question is that I find it very interesting, Francisco, that you have differential accumulation mechanisms in the leaf versus the seed. And so I think that's important because our seed is the ultimate, usage, the utility organ that humans are going to use or are using. And I think the same situation, we see that in some of the crops that can be used as phytoremediation. For example, maize has been used as phytoremediation for cadmium. I'm very interested in this interplay between cadmium and zinc because zinc is important for maize, but cadmium is not. And I think what some studies have found that the cadmium can be stored in the pericarp. I don't know if the same scenario occurs in dicarts like cacao, that the filial tissue or this endosperm can be avoided for accumulation of cadmium, but zinc can be transported. I think that's what probably cacao needs also, right? In case there is a, I'll stop.
Go ahead. He's basically talking about, we missed some of that, but I think it's about how saying competing with it and also accumulating the pericarp, therefore keeping it out of that.
Yes.
Part to consume part.
That's a great question or comment, yeah, or application, because seed development and seed nutrition happens from the mother tissue towards the filial tissue, right? So transport of nutrients, either nutrients like mineral nutrients or glucose or sugars, they all happen from the seed coat, which I think in cereals like maize, it's called the albumin. It stores a high concentration of nutrients and minerals that are going to be then used in the developing plant. So yes, if there is variation and people measure specifically the seed coat cadmium content versus the cotyledon cadmium content, and what I'm kind of hoping potentially that this HMA protein has a function in mobilizing nutrients from the outer layers of the seed into the inner layers of the seed, which we consume. So yes, that would be great, and if there would be a way to like have a protein that only exclusively mobilizes zinc rather than cadmium, even at that very specific level, that would be great for cacao as well, because in cacao, most of the time the seed coat is thrown away, it hasn't been used. So yeah.
But we do have already some evidence as well that most of the cadmium in the seeds is in fact.
Yes, the concentration is way higher.
Yeah, it's concentrated in the seed coat. So that's a good news. I was
Yeah.
trying to understand it better.
Great. First of all, nice talk.
Thank you.
I'm curious if it is known or you know anything about, so the HMA proteins, they are at the plasma membrane level, right?
Yeah.
So how the cadmium is getting through the cell wall or when it is moving through the apoplast, is there anything known about that?
Yeah, so the proteins responsible for moving the minerals into the cytosol are different. You would have, we're looking at like the evidence show that there's more like the activity of other proteins such as NRAP and Z proteins, IRT proteins are moving from the apoplast into the cytosol. And then HMA proteins are moving it from the cytosol into the apoplast. Is that what you're asking? Sorry.
So what I'm wondering is between your cytosol and apoplast,
Yeah.
you have your plasma membrane and the cell wall.
Yes.
So HMA is at your plasma membrane.
Yes.
So how it is coming from apoplast to your cell wall, or from cell wall to your plasma?
So it's in a solute, right? So it's water. and water is moving towards the center of the root. Whenever this protein comes into contact with or for example, loading, right? If you're the enron protein, it's not ATP dependent. It's gonna be able to mobilize zinc into the cell. Sometimes what you're saying, yeah, sometimes the cell extracellular matrix, it's like a sponge, and it also has negative charge. So a lot of times the extracellular matrix, cell walls trap and serve as a storage organ for minerals. And that is also something that the plants can exploit later on if they go through any deficiencies. It's been shown in Arabidopsis that they can loosen their cell wall structure and release metals and minerals that are trapped in that matrix. And then by the action of these enron proteins or zinc proteins, they get put inside the cytosol across the membrane, right? And once it's in there, it can move by the same activity of mass flow towards the seal. And eventually it's going to come into contact with any other, either an HMA3 that's going to store it in the vacuole again, or it's going to get eventually to the xylem parenchyma and get loaded into the vascular tissue.
I have one more
Yes.
question. Is that maybe you showed, but I missed what's the size of the protein that you cloned?
The size,
In terms of like amino
the
acid?
protein on its own, it's 1,000 amino acids. It's like 3,000 base pairs. So yeah, so it's 1,058. It's pretty big. Just amino acids, residues, yeah. So it's a big protein. It has 9 exons. Yeah. Time for a question. Any other questions?
Thank you.
Thank you for that.